Okay, so this is the final, hopefully, statistics portion of this mini class, and so we are finally making our way to null hypothesis significance testing, which is what probably everyone thought statistics was going to be. But it turns out we're at the last stage, because we've been building up sort of basic intuition of how numbers work, how we describe numbers. And now finally we're going to get to be really talking about how do we describe differences between sets of numbers, which is fundamentally what most of us are interested in as biologists. Are two sets of numbers different? So the other thing that null hypothesis significance testing is known as is p-values. And probably doing the homework and during the last statistics course, some of you were getting the sense that maybe we were doing something that seemed a little like p-values. We were computing probabilities of things happening by chance and asking, are these probabilities less than 5%? So really the homework that you did prefigured this, and now we're going to talk about the actual sort of theoretical underpinning. How do we go about evaluating questions like this, asking questions about differences in sets of numbers rigorously. So let's break this this big phrase apart. What is null hypothesis significance testing? Well, first, what is the null hypothesis? Basically, if you remember the very beginning of this course, I talked about how statistics is how we arm ourselves against the all too human tendency to fool ourselves, to see a pattern where none exists. So the null hypothesis is fundamentally the uninteresting explanation that there is no pattern, that it was just random chance, that you're just seeing shapes in the clouds. And so formally in your sort of basic experimental biology perspective, the null hypothesis is that the experimental group is no different than the control group. That these are the same groups, and your treatment was no more or less effective than just adding water to the cells, or adding powdered pixie dust to the mouse chow, or whatever it is. That's the null hypothesis, that any differences are due to chance. Now we contrast the null hypothesis with what's called the alternative hypothesis, the alternate hypothesis. And that's what we hope to see, that there is some measurable difference between the populations in some respect or another. So a little bit of notation, we call the null hypothesis H with a 0 subscript, so null, 0, null hypothesis. So the simplest null hypothesis is that so this is common notation for the mean. So the mean of our experimental group is the same as the mean of our control group. That's sort of the basic, very simple null hypothesis that you've seen in any kind of statistics course that any of you may have taken. And then, in contrast, what's the alternative hypothesis? So, maybe we see that our mice that we're feeding blueberries to live 3 days longer than our mice that aren't being fed blueberries. So do we want to say, on average? So is our alternative hypothesis that the mean of the experimental group is 3 days less than the mean of our, or is 3 days more than the mean of our control group? It turns out no, we don't make a specific prediction. The null hypothesis is the general case that there is something different going on. So usually we would phrase this as just, there's anything different that's not, they're not totally identical. And then the null hypothesis significance testing procedure allows us to evaluate these two possibilities based on the data that we observe, based on the observed difference of 3 days, say for our blueberry versus normal fed mice. So how do we formalize that? That's where we get to a p-value. So what is a p-value? What is a p-value telling us? Fundamentally, the way you want to think about a p-value is very simple. It's exactly what we were calculating before in the last class in the homework. It's this quantity. It's the probability of getting an observation as or more extreme than the one that we obtained, given the null hypothesis. So let's write this a little more pragmatically in the case where we observe that the experimental group lived for 10 months, say, on average, and the control group of mice lived for 8 months on average. So what's our observation? Our observation is that the experimental group was 2 months diverged from the control group. And so we want to know, what's the probability So what's the probability that we would observe a divergence of more than 2? And here I put absolute values, so we're going to go in either direction. Because a priori, again, we don't know that maybe blueberries are shortening life. We don't want to assume that the effect is going to go in one direction or another. That's sort of putting our fingers on the scale. So what's the probability of getting an effect this big or bigger under the null hypothesis that in truth, these two populations are identical? Does that make sense? Anyone have any questions about this definition? I'm playing a little fast and loose with the notation, but we're going to get that tightened up in a little bit when we start talking about the true population versus the sample. Okay, so again, the p-value is strictly how likely is what we see due to chance alone. We use this number, we use the p-value only, and only, never for any other purpose than to tell us how likely is it that we're deceiving ourselves. How likely is it that we're seeing shapes in the clouds, that this is not a real effect and we're squinting too hard at our data and hoping too much that blueberries do something. That is the only purpose of the p-value. So, what is a p-value not? So let's write down in words what a p-value is and what it isn't. So the p-value is that's what a p-value is. How likely is what we see due to chance alone? What is a p-value not? This is important. This is something that people mess up all the time, and we're going to work through how these mistakes happen and why they're really important not to make. P-value is not. P-value is not answering the question, from my data, how likely is there no effect? Or equivalently, from my data, how likely is there to be a real effect? They're sort of the same question, right? these seem like very similar questions, the what is a p-value and what is not a p-value, right? Like, how are these different? Why is one of them correct and one of them a huge error? So remember, we've defined a p-value as the observation as or more extreme given the null hypothesis. So I'm going to shorten this, and we'll write, okay, p-value is the probability of our observations given the null. Make sense? So this is just compact notation for the above. What is this question asking? Does anyone want to translate this first sentence into a probability statement? So this first sentence is asking, what is the probability of the null hypothesis given my observations? The second question is asking, what is the probability of the alternate hypothesis given my observations? Now, remember from our basic probability lecture about how we combine probabilities that these two values are not the same. In fact, they're fundamentally different values. And we had to go through all kinds of hoops to convert this to this. But a lot of people don't necessarily do that when they're thinking about p-values. They read a p-value and they think, ah, it's very likely that there is no effect given what I've seen. That's not what a p-value is. A p-value is how likely is it that what I have seen would exist, would come about due to no effect. So one, you're assuming the exact opposite. So how does this cause problems in reasoning? How does this cause mistakes in biology? And so fundamentally, the real problem here is that the more interesting thing that we would like to know, the question that we would like to answer, is this question. How much should we believe in the alternate hypothesis, given what we've seen? Is our evidence enough to make us think that the alternate is true? What is the probability of the alternate hypothesis, given what we've seen? That's what we really want to know. That's what we're trying to answer as biologists with gathering data. We want to gather more data until this probability becomes big, and we can say, yes, it's true. And so why are we even doing this? Why are we gathering more data until this probability becomes small? So let's actually work out. I mean, these are mathematical statements, and I showed you a mathematical relationship between the two. So let's work out the relationship between this question, which is sort of what we want to know, and this question, which is what we can calculate. All right, so let's just crank through the math. So, again, we really want to know equivalent statements. What's the probability that this effect is real? Or what's the probability that I'm fooling myself? Right, that's what we really care about. So let's ask the fooling ourselves question. So you should all be able to write this down, that I'm going to write down from what we've learned. This is just Bayes' theorem, like we talked about a few lectures ago. So far, so good. Problem is, a lot of people stop here. So remember our example of looking through compounds to try to find an active compound. And we wanted to know how likely is a positive test going to be an actual active compound, given that we know how likely an active compound is to give a positive test. This is exactly the same question. In fact, sort of philosophically, deeply, fundamentally the same question. So all right, I mean, this seems pretty straightforward. This is what we want to know. This is what we know. Now we just have to supply values for P null and P alternative. Sorry, what? What are these quantities, right? Like, what do we mean, the probability of the null hypothesis being true? Like, aren't we scientists? Don't we evaluate probabilities of hypotheses being true given evidence? What could it possibly mean? So these are philosophical terrors. These are the boogeymen that strike fear into the heart of well-meaning statistics instructors everywhere. Because we need to somehow come up with a probability of something being true or false without actually knowing whether it's true or false. But again, if we look to our drug screening example, where we said, okay, well, we don't know how many compounds are really active in my 5,000 compound library of natural extracts from Venezuela. But we say, okay, well, I guess there's probably 20 active compounds. I would be willing to bet there were 20 active compounds. So we said, okay, the probability of an active compound is 20 out of 5,000. And we just cranked through the math from there, and it worked out okay. And you could change this probability a little bit and see how that would change the math that you get. And so fundamentally, again, we just have to make sort of a philosophical commitment. Okay, I think that it's probably likely that the blueberries don't do much for cancer. And so really what we want to manipulate the math to is to get this quantity. So what's the ratio of the probability of the alternate hypothesis given what we see to the null hypothesis given what we see? How much more likely is it that the null hypothesis, that the alternate hypothesis explains our data? So if we use this and crank through a little bit, we can get an equivalent definition of this. Okay, so this cleaned things up a little bit. This is called the Bayes factor. How much more likely is one explanation than the other? And okay, so we can do all right with this. We know how to calculate this, we know how to calculate this. These are p-values that we know how to deal with. And then these are, again, these sort of prior probabilities of something being true or false. And again, that seems weird, but this is really just the mathematical version of the statement, extraordinary claims require extraordinary evidence. So extraordinary claims, like ESP exists, I am a mental telepath and I can read your mind. That's an extraordinary claim. There is a very low ratio between my sort of prior probabilities of this being true and false. Like 1/1,000,000% I would believe that someone has ESP. Now that doesn't mean that you couldn't come up with an experiment that's going to convince me that ESP is real, but it's just going to have to be a really good experiment. And that really good experiment would give you a huge ratio between the observed, the probability of the observations given the truth that ESP happens versus the probability of the observations being just by chance. So right, to convince me that ESP is real, you're going to have to really show me seriously that your experiment was not confounded by chance. In a way that you're not going to seriously have to show me that, Taxol keeps a different kind of tumor at bay that you haven't previously tested. Like, these are qualitatively different scientific endeavors. proving that ESP is real versus proving that Taxol works against yet another cancer. And fundamentally, this has to do with thinking about, okay, how likely is this thing to be true? How extraordinary are my claims? Now, unfortunately, all right, so number one, these numbers are just sort of made up. They come from, they exist, they have to be real, but we can't calculate them. And even worse, this probability of our observations given the alternative hypothesis is also really hard to calculate in most cases and involves integrals for one. Because if you remember, the alternative hypothesis is just this huge class of anything could be true as long as they're not the same. So this is really hard to calculate. So unfortunately, kind of what we're left with, the only thing that's easy to calculate is the probability of the observations given the null. And, you know, that's too bad. But the most pragmatic thing to do is say, okay, yeah, these are philosophically terrifying. This is really hard math. This we can do, so let's just deal with that. You know, okay, it doesn't answer the question we want it to answer. That's fine. But you have to understand that. You have to understand what a p-value really is telling you, which is merely the chance of your observations arising due to random effects alone. And if you don't overinterpret your p-value, then you're fine. So really, again, we'd like to answer this question, this Bayes factor. It's really hard to answer this question, so we're just not going to. We're going to answer a question that we can answer rather than the one we want to answer. It's sort of unsatisfying. Nevertheless, that's what we do, because that's what we can do. So, we're back to our p-value. We're not going to calculate Bayes factors. We're not going to ask about the prior odds of ESP. We're just going to ask the probability of an observation as or more extreme given the null hypothesis. And if this probability is suitably small, if it's a tiny chance that what we see would arise just by random rolls of the dice, we say that we reject the null hypothesis. That yes, the null hypothesis can only explain the data 5% of the time. And that's enough that we're saying that is not true. We reject the null hypothesis. And another way of saying that we reject the null hypothesis is saying that the result is significantly unlikely to be due to chance alone. So that's where this significance comes from. We're talking about likelihood of chance alone. Now, note, and this is really important, this isn't just sort of linguistic argle-bargle. You never accept the null hypothesis. You merely fail to reject it. What's the difference between accepting the null and failing to reject the null? So the problem is that failure to reject the null hypothesis is not evidence for the null hypothesis. Just because you couldn't conclusively say it wasn't due to chance doesn't mean that it might have been due to chance. Those are not logically equivalent. For example, in our cancer blueberries example, where we saw that there was this 20% difference in survival and cancer rate between mice fed blueberries and mice not fed blueberries, and we said, okay, we cannot reject the null hypothesis. We failed to reject the null hypothesis. It was easy to explain the difference we saw due to chance alone. But that doesn't mean that there was no effect. We couldn't convince ourselves that there was an effect, but that does not mean that there was convincingly no effect. This seems like sort of playing around with language. Why am I belaboring this point that we don't accept the null hypothesis, that failure to reject the null is not evidence for the null? The reason I'm belaboring this point is that people die every day because of this mistake. So, I don't know if you guys know about the history of right turn on red laws. Does anyone know when right turn on red laws came around in the US? So it was in the height of the Jimmy Carter era when there was the oil prices were going up and up and up and everyone was waiting in line for gasoline at the pump. You've probably seen those black and white pictures of the lines for the gas, and and then Jimmy Carter didn't get reelected because no one liked gas. And so people were thinking, well, how can we save gasoline? Maybe if you didn't have to stop so long at a stoplight, you could just, come to a halt, and then turn right on red. Seems like a totally reasonable thing to do. We're totally used to right on red. But that was a relatively recent innovation. And people were worried at the time that obviously right on red has this big problem and that there are pedestrians crossing the street at the same time that you're trying to turn right on red. And so, you wouldn't want to enact a national right on red law or even a state right on red law unless you were fairly convinced that pedestrians weren't going to get plastered in intersections every day because of this new law. And so, several studies looked at the rate of accidents in right turn on red states or towns that had right turn on red zones, and they saw an insignificant effect on pedestrian fatalities. They failed to reject the null hypothesis. And so they said, well, we don't have evidence that this is a big effect, so we're going to assume that the effect is zero. They accepted the null hypothesis based on their observations. That was absolutely wrong. These were tiny studies. These were crappy studies, the equivalent of doing this blueberry thing with 6 mice. And if they had done better studies, they would have seen that the effect was real, not huge, but real. And if they had a big enough study, they would have seen a statistically significant effect. So again, not having a significant effect doesn't mean there is no effect. The best guess was still that there was this true, slight positive effect of getting plastered in an intersection. They just had done a crummy study. And so that's why it is fundamentally important to distinguish between not being able to reject the null hypothesis, which is either due to the null hypothesis being true or your study being crappy, and being able to convincingly say to yourself, no, I did a good enough study that I can start to think that maybe the null hypothesis is true. These are two fundamentally different scenarios, and in many different circumstances, not just right turn on red. These are life and death decisions that get made wrong all the time. So fundamentally, we need to think about this. All right, now who got all the way done with the homework, the cancer mouse homework from the last statistics course? Anyone? Okay, a couple hands. So it's really important to do that because that's the beginning of starting to ask these questions. The very end of the homework was, okay, how many mice are we going to have to do this study of if we really want to be able to convincingly detect this small effect? And it turned out you needed to use a huge number of mice in order to see what seemed like, a 20% effect. That's pretty big. You had like 150 mice in your study in order, if that effect was observed, to really be able to convincingly say it wasn't due to chance. So these kind of errors, false non-rejection of the null hypothesis, are really problematic. And usually, you're only going to shoot yourself in the foot. You're saying, oh, okay, well, we didn't see blueberries were effective, then you lost out on the opportunity to patent blueberry juice and make $1 million and save a bunch of people's lives. Like, somehow that's more okay than this right turn on red scenario, where by saying something wasn't a problem, they actually wound up killing a bunch of people every day, and now that's never going to change because we're just used to it. Just another factor of carnage on the road. Okay, so that's fun. It's not like someone dear to me died at an intersection. It's just, an example of
I don't say that. Because we don't have it on video.
Oh, yeah. Okay, so that's one class of errors that people make all the time by misinterpreting what a p-value means. What's the other class of errors that people make? So the truth is that almost everything has some effect. It's almost never true that these things are going to be exactly equal. And sometimes the effect is just due to random fluctuations, and the p-value can distinguish that. Sometimes the effect is due to confounding factors. Maybe blueberry chow is a different color, and mice that look at blue food eat a little bit less food, and it was the eating less food, not the blueberries. There's all these different reasons that might make these groups not completely identical. And so if you do a big enough study, you can detect, oh yeah, these groups are totally not identical. They're different. Without having to really say anything about whether they're meaningfully different. And so, you know, these kind of errors don't typically kill people, but these errors waste billions of dollars every year. All the time, new drugs come out that are advertised as being significantly better than the current baseline best therapy. So what do they mean? So, you know, when I as a human say something is significantly better, I mean, it's a lot better, it's bigger, the effect is big, it is significantly better. You will live significantly many more months on this drug than that drug. What they mean is, again, the definition of the null hypothesis significance testing. They mean the result is significantly unlikely to have come about due to chance alone. Significantly unlikely to come about due to chance alone is very different than significantly bigger. You can have a tiny, tiny effect that if you measure it very carefully will be significantly unlikely to come about due to chance alone. So that's why we have these $60,000 cancer treatments that on average will buy a patient one more week of life. And insurance companies are obliged to pay for them because they're FDA approved, they are significantly better than the baseline therapy. But in reality, for no individual patient is the drug going to make any difference. So this is the other class of errors that people make with p-values. So again, what are the two errors? Error number one is assuming that a non-significant result means that the null hypothesis is true. Error number two is assuming that a significant result means the alternative hypothesis is interesting. All right, so p-values are kind of useless when really thinking about what you should do, how should you behave, what drugs should you take, what laws should you pass. P-values are a tiny, tiny part of the puzzle. And the rest of the puzzle is being sort of wise about how to interpret p-values and how to use them. So again, p-values are really important. They're your important bulwark against thinking that you see something that you really don't see, that doesn't really exist. But again, that is all that they are. So if you come out of this class with nothing else, remember that a p-value is the probability of an observation as or more extreme given the null hypothesis. It is nothing more than how likely are these results due to come, how likely is observing these results due to chance. It does not tell you anything about how likely the alternative explanation is or whether or not you should accept the null hypothesis. All right. So, I mean, okay, we're going to do experimental biology. We're going to have to calculate p-values and not just cower in fear from them. So let's now talk about how we calculate p-values. All right, so fundamentally, what do we need to know to calculate the probability of an observation given the null hypothesis? What do we need to know to calculate the probability of anything? How did we calculate, how did we start to ask questions about how likely 6 exons or more was? What did we need to know? We needed to know the distribution of exon counts across the genome. We needed to see the histogram in front of us and say, you know, here are the exon counts. So these are supposed to be straight lines. So here's the histogram of number of exons for every gene in the genome. How likely, what is the probability of getting 6 exons or more? You look at that area and you say, okay, that's the probability. So fundamentally, to ask questions about probability, we need to know the distribution. We need to know the histogram of the thing that we're interested in. All right, so where does that leave us with this null hypothesis significance testing? We need to know the distribution of the thing that we're measuring when the null hypothesis is true. So that seems a little weird. What exactly is that? So I'm not going to draw histograms anymore, I'm just going to draw smooth lines. Again, in the first class we talked about how a histogram is sort of, or a smooth line is kind of a limiting case of a histogram. So, imagine that we just fed 2 different groups of mice normal chow all the time. Well, group A would have normal chow and group B would have normal chow. They're obviously going to have different lifespans, right? The lifespans of group A and group B are not going to be identical always. There's going to be some distribution of sometimes group A is going to live a little longer, sometimes group B is going to live a little longer. So if we could repeat those experiments infinitely many times with infinitely many mice, and we had plenty of money and we just wanted to keep doing control experiments, we would get a distribution of the lifespans of mice, or sorry, just a distribution of the average lifespan of, say, a group of 6 mice that we could build, we could build up a histogram. So, we take 6 mice, look at their average lifespan. We would say, okay, average lifespan is here. Then we take another 6 mice, feed them the same chow, look at their average lifespan. It would be here. And we can do this again and again and again, more and more and more groups of 6 mice. And eventually we could build ourselves a histogram of what lifespans of mice look like with no perturbation, no nothing. So fundamentally, that would be then the null distribution for average lifespan of a group of 6 mice, assuming that there is no effect of whatever you're feeding them. Does that make sense? Our statistic of interest is the average lifespan of a group of 6, and we want to know how is this distribution going or how are these numbers likely to fall out due to chance alone? Just repeating our experiment. And then if we do, one time we actually feed our group of 6 mice some blueberries, and we see a lifespan over here. Say, okay, now we can get somewhere. This distribution, this curve tells us the probability due to the probability, the sort of spread in lifespans that you would expect due to chance alone. Sometimes 6 mice are going to live less long, sometimes they're going to live more long due to chance alone. And we've built up this distribution by this hugely expensive process of taking hundreds and thousands of groups of 6 mice and seeing how long they live. And now that we've done that, we get another group of 6 mice where we have fiddled with them somehow. You see, this is out here. How likely is this observation due to chance alone? And we say, okay, well, if the observation was here, it'd be pretty likely to be due to chance alone. So remember, the p-value is what is the probability of an observation as extreme or more due to chance alone. So let me draw this out a little further. So we have some mean lifespan of unperturbed animals. We have our observed lifespan of these perturbed animals. So we want to know what's the probability of this observation given the null hypothesis that the perturbation was merely chance effects alone. So that's easy. Okay. This area here is the probability of an observation this big or bigger due to chance alone. And remember, we generally want to look in both directions. So this is one tail of the distribution. We'll say, okay, we want to go out this far or out that far, same distance. So these red areas, the area of the null distribution that's past our observation in any direction, is fundamentally the p-value. It's the probability of seeing something this crazy due to chance alone. Now remember, if we had infinite resources, we could keep sampling groups of 6 mice, and probably at some point we would get, a few groups of 6 mice all the way out here. So it's not like you're never ever going to see something as crazily different as you saw in your blueberry mice. So we really want to know, how likely is that to have been something that we would just happen by random repetition? All right. So this then is the null distribution. gives us the probability that the observation is some value given the null distribution. And then we can look at the area under the curve to ask about the probability of the null distribution being bigger or smaller. Anyone have any questions about this?
Why do you have to do the two sides? Can you do a single-sided?
Yeah, so if we just look at this area, that would be the one-tailed p-value. And if we look at these two areas, this would be the two-tailed p-value. Now, most of the time, this is going to be symmetric. So you say, well, our two-tailed p-value is going to be twice as crummy as our one-tailed p-value. But unless you know that your observation could never be below the mean, then the two-tailed p-value is not useful. Because again, the null hypothesis is that, the one-tailed p-value is not useful. The null hypothesis is that what you're looking at is coming from random sampling alone. And so if there's nothing about your random sampling procedure that constrains your samples to only come from this side, then you're not calculating the p-value correctly. Because remember, the p-value is the probability under the null hypothesis of getting data out here. So unless your experimental setup is such as you could never conceivably get data over on that tail, then you have to use a two-tailed p-value to be fair. And again, with these blueberry mice, it's totally conceivable that the blueberry mice could have a shorter life. So that's why, in general, you want to use the two-tailed test, calculate a two-tailed p-value, unless you have really convincing reason to not. And convincing reason is not, oh, our observation was big, so that's circular reason. Okay, so how do we obtain this null distribution? Right, I described one procedure, which is actually fundamentally the only real way to go about getting a null distribution. We sample repeatedly from the true population of all possible white mice. We take six mice at a time repeatedly and look at their distribution of the lifespans, of the average lifespan of those group of 6 mice. That is fundamentally what the null distribution is, and the only way we could ever get it. So how would we do this? We would just do our experiment over and over and over again with the negative controls. Do 10,000 different negative controls. Build this distribution, and then with our one experimental sample, see where it falls on that distribution. And that gives us exactly the p-value. So this is conceptually extraordinarily straightforward, even though it's sort of experimentally unfeasible. Now sometimes we actually have direct access to the true population. Remember, in the exon count example, we know all of the protein coatings in the genome, protein coding genes in the genome. That is the true population. We could, so remember this example was, we had a sample of genes. We had some genes that came down from ChIP-seq, and they seemed to have more low exon genes than you would expect they seem to have too many genes that have too few exons. You say, okay, how often would this be due to chance alone if we just randomly grabbed sets of 20 protein coding genes? So because we have the full true population, we have all of the protein coding genes in the genome in this file in front of us, we could build up the true null distribution. We could tell the computer, take every single possible unique set of 20 genes, calculate the counts of the exons, and then that would be the null distribution. So conceivably we could do this. It would take a long time. There's a lot of protein coding genes, and so there's a lot of different possible combinations of 20 that you could pick. But conceptually, because we have access to the null distribution, the access to the true population, we could empirically build the true, absolute, actual null distribution of the statistic of interest. Okay, so that would be computationally unfeasible. So what do we do instead? We just randomly took a few different possible groups of 20 genes and assumed that their distribution would look quite a bit like the distribution of exon counts of groups of 20 genes if we did every single group exhaustively. So that's called a Monte Carlo method, where we just take random samples and assume that they're going to look something like this true null distribution. Now, sometimes when we have the true population, again with all the genes in the genome, we can actually use math to solve for what the shape of the null distribution would look like without having to tell the computer, okay, give me every possible sample of 20. And so in this case, with the genome counts, we could have used math. But instead we just said, oh, computer, take us some samples. That's less likely to be wrong than me trying to work through the math. We saw that with the Monty Hall example, that working through the math is sometimes problematic, but it's rarely problematic to tell a computer to grab you 20 samples a bunch of times and make a histogram of it. Okay. So, except for these rare cases where we actually do have true access to the null distribution or have unlimited funds